Yeah, in this presentation, let's understand how to identify carbohydrate. Coming to physical properties, state is liquid, carbohydrates are colorless, odorless compounds, and reaction to litmus is neutral. Coming to the chemical test which we need to perform, these are the battery of tests we have to perform to identify the carbohydrate. First one is the Molish test where we get to see formation of violet ring at the junction of two liquids, and this indicates the presence of carbohydrate. Now this is a general test for carbohydrate. What's the principle behind this test? Carbohydrates when treated with concentrated sulfuric acid, they undergo dehydration to form furfural derivatives. Now these furfural derivatives further condense with phenolic compounds like alpha naphthol to form violet colored complex, which is seen as ring at the junction of the two liquids. Now the next test is iodine test, where we get to see no color change, which indicates polysaccharides are absent and the compound or the carbohydrate may be mono or disaccharide. Now what's the principle behind this test? Now starch is composed of two compounds, such amylose and amylopectin. Now this amylose has got spherical structure, and this iodine when added gets absorbed at the branch points of the iodine, and this complex gives a intense blue color. So only when polysaccharides are present there is appearance of blue color, and when there is no appearance of blue color it indicates polysaccharides are absent and the compound may be mono or disaccharide. So the next test we need to do is the Benedict's test, and we get to see formation of colored precipitate. So depending upon the concentration of the carbohydrate present in the given solution, the color varies from green, yellow, orange and red. Now this Benedict's test is a semi-quantitative test. It is both qualitative and quantitative. Quantification is based on the different colored precipitate formation. For example, when green colored precipitate is formed, it indicates 0.5% of the carbohydrate in the given sample. Yellow indicates 1%, orange indicates 1.5%, and red indicates 2% of the reducing sugar present in the sample. Now what's the principle behind this test? Reducing sugar in hot alkaline condition tautomerize to form NADOLs, and these NADOLs are powerful reducing agents. So they reduce copper sulfate of Benedict's reagent to cuprous oxide, which gets further converted to cuprous hydroxide, forming different colored precipitate like green, yellow, orange and red depending upon the concentration of the reducing sugar. So the next test we need to perform is Barfoot's test to know whether this reducing carbohydrate is a monosaccharide or not. So this Barfoot's test is mainly done to identify whether the carbohydrate is monosaccharide or not. So if there is a formation of a red granular precipitate at the bottom of the test tube, it indicates the presence of monosaccharide. What's the principle behind this test? Monosaccharides with free aldehyde or keto group can reduce cupric ions to cuprous oxide in acidic condition. So when this test is positive, now we got to know that this is a monosaccharide. Now we have to identify which type of monosaccharide is this, whether it is aldose sugar or a keto sugar. So the next test we need to perform is Selivanov's test. If there is formation of cherry red color, it indicates keto sugar. In this sample there is no formation of cherry red color, so it indicates keto sugar is absent and aldose sugar is present, and the most common aldose sugar is glucose. What's the principle behind this test? So resorcinol, which is present in Selivanov's reagent, react with furfural derivative to form cherry red colored complex. Here, so there is no formation of cherry red color. So this indicates keto sugar is absent and it is aldose. Now how to report this sample? So the given solution is carbohydrate reducing monosaccharide aldose sugar or aldose glucose. Now what are the properties, functions and the structure of glucose? So structure of glucose, it is a aldohexose and it occurs in beta D glucopyranose form. So what are the properties of glucose? Now glucose can be oxidized. Glucose, as you all know, it has got 6 carbon atom. When the first carbon atom is oxidized, it results in the formation of gluconic acid. When the last carbon atom of glucose is oxidized, form glucuronic acid. And when both first and last carbon atoms are oxidized, it forms saccharic acid, glucosaccharic acid. Now glucose can be reduced to sorbitol. Glucose forms broomstick shaped ozone crystal. Now this is how the crystal looks when we observe under microscope. Next is replacement of the OH group at the second carbon atom of glucose forms amino sugar. Glucose is reducing agent because it reduces it answers Benedict's test positive. Now glucose is dextrorotatory with an activity of plus 52.5. Coming to functions of glucose, now glucose acts as a primary source of energy for brain and RBC, and also acts as a source of energy for all the peripheral tissues by getting oxidized in glycolytic pathway and further getting oxidized in TCA cycle. Now, glucuronic acid, which is formed by oxidation of last carbon atom of glucose, is involved in detoxification reactions, detoxification of harmful compounds and also foreign substances, and it is a component of glycosaminoglycans, mainly the hyaluronic acid and chondroitin sulfate. Glucosamine, which is found by removal of OH group at position 2 with replacement of amino group. It is a component of glycoprotein, and it is also a component of glycosaminoglycans, mainly hyaluronic acid and heparin. Now, glucose through HMP pathway, it provides pentose sugar for the formation of nucleic acids, DNA and RNA. Mainly the ribose sugar is involved in the formation of DNA and RNA. And through HMP pathway, it provides NADPH, which is used for reductive biosynthesis of fatty acids, cholesterols and steroid hormone. Coming to the clinical significance of glucose. So when large amount of glucose is present in circulation, it can this condition is known as hyperglycemia, and it can predispose to the development of type 2 diabetes mellitus, because glucose is under the hormonal regulation of insulin and glucagon. So when there is decreased insulin, so glucose is not taken up by the cells, especially the especially by the muscle cells and the adipose tissue. So it accumulates in the blood and results predisposing the patient to type 2 diabetes mellitus. So when it exceeds 180 milligram per deciliter, it gets excreted in urine, and this condition is known as glycosuria. So glucose can be reduced to sorbitol. So when there is excess accumulation of glucose in the circulation, it gets reduced to sorbitol, and this can predispose the diabetic patient to development of diabetic retinopathy and neuropathy. So this is the structure of the glucose, which is present in the pyranose form, and to the anomeric carbon atom, with at the anomeric carbon atom two different isomers, stereoisomers can be formed, that is alpha D glucose and beta D glucose. And because of this formation of alpha D and beta D glucose, it the mutarotation property of glucose is mainly because of this alpha D and beta D glucose. So this is all about identification of carbohydrate, in particular glucose, and this presentation was done by Dr. Preeti, Professor of Biochemistry, JJ Medical College, Davangere. Now here are the links for analysis of abnormal constituents in urine, different biochemistry practical links are there. You can access the videos by clicking on this links. Thank you.